Hi, my name is Ran. I work as a scientist in the Department of Surgery at University of Wisconsin-Madison. My presentation title is Variation of Resident Microbiota in Response to Mucosal Healing in the Larynx. Voice disorders are among the most common communication disorders and estimated to affect 3 to 9% of Americans annually. These disorders are often associated with laryngeal inflammation secondary to epithelial injuries following phonotrauma, surgical resection, radiation, and various infectious processes. Bacterial dysbiosis, a disturbance in the microbial community structure, is implicated in a variety of inflammatory disease etiologies in mucosal systems. However, understanding of laryngeal microbiota and their role in mucosal inflammation remains limited. In this study, we investigated the variation of laryngeal microbiota in response to wound throughout the healing of laryngeal mucosa in a naphthalene-induced epithelial injury mouse model, where normal 6-week-old mice were injected with naphthalene and intraperitoneally and monitored for 28 days. Larynges were collected at 1, 3, 7, 14, and 28 days post-injury aseptically, and DNA was extracted from this tissue. V4 region of 6nes rRNA gene was amplified and sequenced on a MiSeq platform, and data were analyzed with QIIME2 and visualized in RStudio. Relative abundance of laryngeal microbiota at different time points after injury was characterized and compared between injured and non-injured group at each time point. Significant difference in bacterial relative abundance between the two groups were observed at 1, 3, and 7 dpi. On 1 dpi, the relative abundance of Streptococcus and Pseudomonas were significantly increased in injured mice, while that of Lactobacillus and Sphingomonas were significantly decreased in the injured mice. On 3 dpi, the relative abundance of Pseudomonas remained higher in injured mice, while the relative abundance of Streptococcus became similar in both groups. On 7 dpi, Oxalobacteria became dominant in the injured group, but little difference was observed on 14 and 28 dpi. Within group diversity was estimated through alpha diversity, which was significantly increased in 1 day post-injury, but decreased in 7 day post-injury. No significant difference was observed in alpha diversity at other time points. Beta diversity analysis suggested that bacterial community structure was significantly altered on 1, 3, 7, and 14 day post-injury, but not on 28 day post-injury. Differentially abundant analysis revealed that Pseudomonas were significantly enriched in injured group at 1 and 3 day post-injury, while Lactobacillus abundance is significantly reduced on 1 and 3 day post-injury. The abundance of Streptococcus also seemed to play a role in wound healing process, where it was enriched in injured group on 1 day post-injury and subsided at later time points. Conclusion: microbial relative abundance changed in response to mucosal wound throughout healing process, especially at 1, 3, and 7 day post-injury. Most responsive taxa were Pseudomonas and Streptococcus, which are potentially pathogenic bacteria, and Lactobacillus and Sphingomonas, which are potentially beneficial bacteria. Microbial diversity and abundance varied temporally through healing. However, larger sample size is needed to prove. To move forward, we would like to define the role of commensal microbiota in acute wound healing of laryngeal mucosa by comparing host biology between germ-free and conventionally raised mice. We would also like to study host responses to monocolonization of wound responsive strains using germ-free mice, such as colonizing germ-free mice using the potential candidates that are found in our current study, including Streptococcus, Lactobacillus, Pseudomonas, and Sphingomonas.